So I cut out all my gloom and doom slides about coronavirus because this audience probably doesn't need that scare. But John and I and others have been trying to now convince people that this is not a once in 100-year event, and this kind of chronology of pandemics, I think, really kind of graphically drives that home that nature is trying to tell us something, right? The pace of these pandemics are accelerating, and like hurricanes and droughts that used to be once in 100-year events, the landscape has changed, right? And so we cannot just say, okay, well, let's leave this for another generation to deal with. But things that we're doing and you guys are doing, I think, are going to be critical. And we need to convince our elected representatives not to look at this in the rearview mirror, but be proactive on these kinds of pandemic prevention issues. So at CAMP, we primarily focus on SARS-CoV-2 with programs in flavy and hemorrhagic fever viruses. And so here's a list of targets that we're going after and the stages that they're in. I'm going to go through them in this swim lane slide and give you an overview and then give you some data. And so just to kind of orient you, the ends of these arrows are aspirational. That's where we hope to be at the end of year 3. Obviously, for those of you who have done drug discovery, know that probably 80% of these are going to be wrong. But again, this is kind of, if everything goes right, this is where the trajectory is. The yellow triangles here are the current stage of these programs. So one of our most advanced programs I'll tell you a little about is the MPRO inhibitor. We have 2 other programs against proteases, a non-covalent MPRO and a PL-PRO. These are not looking as promising for where we are. So again, these are things that we'd be eager to kind of start working with other folks on to see if we can't join forces. We have an oral remdesivir, as was just talked about, but that's stopped. Some of it stopped because of the science, but some of it stopped because the nuke space is heavily covered by IP, and so our oral remdesivir is covered by Gilead's current patent and their current oral remdesivir version of things. And so we didn't think that there was a commercially viable path to this forward. We do have other RDRP inhibitors that we are moving forward. And then I think early stage, but exciting non-nuke inhibitors for RDRP, specifically allosteric inhibitors and inhibitors that break up the RDRP complex. Project 3 has a couple of what we considered druggable targets. And druggable means that there's precedent for drugging that class of targets. We have a helicase inhibitor. We've now killed a capsid inhibitor program. We weren't able to get good enough efficacy. We have an E-protein inhibitor, Stuart Lipton and is working on, and Mike Farzan's working on some entry inhibitor. And then Project 4 is kind of the non-druggable or kind of crazy bucket. So NSP1, which has no enzymatic activity at all. We're trying to use various assays and various new chemistries to go after this protein. And then there are 2 other projects that target the viral RNA. And again, obviously, if you want to target broad spectrum or have a spectrum platform for a RNA virus, any RNA virus, if you could target the RNA, that would be really, I think, a novel and paradigm shifting strategy. Project 5 is our Flavies project. So this is with Sarah Cherry and Mike Diamond. And we have a pretty advanced RDRP inhibitor for dengue. My lab is working on a NS4A inhibitor for Zika, and Mike is trying to target NS1, which again is a non-enzymatic protein. This is kind of a hard to drug target, but they're making some interesting progress there. And then finally, looking at hemorrhagic fever viruses, we have a program on Ebola, Lassa, a nucleocapsid inhibitor for Lassa, and endo inhibitor based on derivatives of baloxavir to block the capsid mechanism. And I'll touch on some of these. And if I didn't touch on them, I'm happy to talk about programs that there may be some overlap on in the post slide session of this seminar. So these are supported these programs are supported by 5 technology platforms. The first 3 are based out of Caliber. And so for those of you who don't know, Caliber is the drug discovery arm of Scripps, and it's kind of a fully contained unit that can go from high throughput screening into phase 2A. A lot of ex-pharma folks have moved over there. And so really it's you know, pretty well integrated pipeline, and they are absolutely open to collaborations at any level, from HTS to MedChem to ADME. So if you need some firepower there, I'm happy to make a connection. John Ciajarro is running our animal model core, and Ian Wilson, Andrew Ward, and Stefano Forley run our structural and computational biology core. So jumping into some of the more target-driven approaches and the molecules that we have, as I mentioned, PL-Pro, M-Pro, RDRP, and helicase, capsid entry, and E-protein and viral RNA. So really kind of targeting various aspects of the life cycle with direct-acting antivirals. And so I'm going to tell you stories around first CL-Pro or M-Pro and RDRP. So one of our most advanced compounds is this main protease inhibitor. As you heard, is pretty highly conserved across alpha and beta coronaviruses. And what we did was use a library, a repurposing library called Reframe, and has about 12,000 known drugs. It's the biggest repurposing library. The Gates Foundation gave Caliber about $20 million to make it, and about half of the 12,000 are drugs that, you know, we had to jump into the patent literature, figure out what they were, and then resynthesize. So they're not commercially available. This library is open to screen for anybody, as long as you make the data public. And so again, if you're interested, reach out to me, and I can do some matchmaking for you to get access to this library. So GC376, the known protease inhibitor that came out of the Reframe library, and that's where we started doing our SAR, and you can see that there was an increase in potency across the derivatives until we got to CMX990, which is right now, depending on the cell line you use, either 36 or 286 nanomolar EC50, and our current lead. So this is a covalent inhibitor, and so after we did some SAR on this and some ADME, where we were able to show that BID dosing got us pretty squarely over the EC90 with this drug. And so we decided to go into the in vivo space. And so this is just a mouse model that John has of an H2 transgenic with SARS-CoV-2. And we tested molnupiravir, Paxlovid, and this CMX990, our internal derivative. We had one mouse, kind of an outlier, kind of acting wonky in both the Paxlovid and the our drug, but for the most part, what we found is that this is viral titer. For some reason, we saw complete rescue in weight loss. We had some funky data on viral titer. but we're doing this again, but we're fairly confident that this molecule is equipotent to the Paxlovid and maybe even better. And I'll explain why we think it's better going forward. We're talking about broad spectrum efficacy. These molecules are the series of the CMX 990. We're hitting alpha and beta coronaviruses and most delta, all delta, and then some gamma, which is a much different spectrum than the Pfizer compound and some other known protease inhibitors. So we're fairly confident that we have hit upon a broad spectrum mechanism of activity here, and it actually seems to be having activity on other viruses that have proteases, including rhino and polio. And so this is an interesting mechanism that we think we can perhaps convert into a broader spectrum molecule on this. So this data was generated by Nick Heaton at Duke University. And so before I switch gears, the king of the hill right now is the Pfizer drug. I think the one big drawback of the Pfizer drug it needs a ritonavir boost. And so the drug-drug interaction list is, I think, 3 or 4 pages long. The line, very small print. The last time I checked, CMX 990 has better potency, better clearance, better protein binding, and better oral bioavailability. So we believe that based on the calculations on human predictions, we can do right now a BID with no boost, and that thereby alleviating kind of these drug-drug interactions. We're hoping to get to a once a day, but we'll see if we're able to get there in the time we have left. So switching gears to our nukes, and so what we did was took a two-pronged strategy. This is work we've done with Matthias Gote. And so the Caliber has a nucleoside library, again, about 700 nukes available to screen with anybody. And so that was screened against SARS-CoV-2 and a heliase-2 replication assay. And then they were converted into triphosphates and counter-screened in, Matthias's biochemical RDRP assay, which is illustrated here. And so we have right now 2 different analogs that work in the micromolar or submicromolar EC50 range. And we've made new analogs with better plasma stability. And what we're hoping to do is get these into in vivo proof of concept by the end of the grant. But these are novel nukes, free and clear of the IP space that's covered by Gilead and Emory and others. It's a very shrinking space, so nukes can be very challenging to develop. But we have a couple of that look fairly interesting, and hopefully, you'll be seeing some nice in vivo proof of concept data in the next couple of months. So I won't have time to talk about our non-nuke inhibitors, but what I will do is tell you how we got there, and this is the same approach that we use for the helicase inhibitors. So I'm going to skip this and jump to the helicase inhibitors. Essentially, we have a helicase assay that we run a high throughput screen on, which is pretty standard. But the other thing we did was we worked with Chris Parker and Ben Cravat to use a human proteomics approach, which essentially are these scout libraries or fully functionalized fragments. They call them FFFs. So there's a small molecule fragment, there's a photoreactive group, and there's an alkyne handle that you can use to do chemoproteomics. And essentially what you do is you transfect, in this case, helicase, but any protein. You add your FFF fragment, and you just look for binding plus minus the transfection, right? Because you can pull things down using this alkyne handle. You can quickly look for binding, right? So this is essentially scouring the protein that you're interested in and looking for binding pockets. And those binding pockets then can be used to elaborate either these FFF fragments or to come up with new drugs. And so what we were able to find is 3 different fragments from Chris and Ben's library that bound by pull down. And then if you put it in the helicase assay, seem to inhibit helicase. Not great, right? These are highly promiscuous, dirty fragments. They're not meant to be drugs. But the fact that you have binding and the fact that you have activity means that you found a pocket that can either be drugged through a competition assay or taking the FFF fragments and turning them into actual more selective drug-like molecule. And so that's exactly what we're doing now with these 3 compounds. What we found is that the binding pocket here is in the 5' phosphate RNA binding pocket of all of these drugs. So they all tend to find the same pocket here, and now we're elaborating this into kind of turning these fragments into drug-like molecules to increase their affinity. Similarly with the RDRP, this is a similar strategy making. We have 2 molecules in the RDRP. Again, Chris is very happy to work with you, and he does a lot of collaboration. So if you have something that you think could benefit from this technology, again, happy to make a connection for you. So switching gears to the RNA targeting aspect, this is work done by Matt Disney over at University of Florida. The idea here is twofold, right? So this is called a ribotac, which is like a protac except for RNA. So a protac will recruit an E3 ligase to your target of interest and then drive its degradation. Here, what we're doing is we're finding drugs that bind, small molecules that bind RNAs, right? And so this is first an RNA binder, and then we add a recruiter. In this case, it recruits it to RNA cell, which drives degradation. And so Matt's set up a library that is enriched for things that are likely to bind RNA, about 4,000 drug library that he screened. And so far we have 4 binders that in and of themselves at single-digit micromolar can bind RNA and block replication, okay? So right now what we're doing is that we're converting these into ribotac, so adding this RNA cell recruiting domain, and we're hoping that we can convert this from the micromolar activity down to triple or double-digit nanomolar using this approach, and really think that, you know, if we can establish this kind of paradigm, it can be used for almost any RNA virus, right? A very selective binding, if we can find a highly conserved region in the RNA virus to target and then target it for degradation, we start getting broad spectrum activity against different families, and really targeting kind of the guts of the virus here to drive antiviral activity. So the other project that we're working on is on PPMO. So Hong 10 is leading this out of Oregon, and so essentially PPMOs are morpholinos. Okay, they're like RNAi nucleotides that you add a cell-penetrating peptide to and a linker, and it targets the RNA for degradation, or it blocks translation of the RNA. So we've been using this for a while as a tool for flu to validate flu host targets and now SARS-CoV-2 host targets. But the thought here was that can you make these into an antiviral therapy? And so the data is actually better than we expected. So this would be an intranasal drug. You could imagine taking these either prophylactically or post-exposure, where you just squirt it up your nose, gets into kind of your upper respiratory tract, nasal epithelium. And what we found is that these PPMOs, when you deliver them intranasally, they work as good or better than molnupiravir, right? So, you know, we dosed them twice and then looked at what the viral titers were. And I think this is an interesting outpatient for formulation opportunity for people that can want to do prep or post exposure, especially, you know, if you have a condition that makes you more susceptible and you're getting on an airplane or going to concert or what have you. So we're pretty excited about this. and really kind of this was in that kind of bucket of crazy projects that we submitted, but every once and again, when you swing for the fences, something ends up working. So we're trying to get a formulation here to move it into a kind of a better intranasal formulation for delivery and moving into NHP in clinical trials. So I'm going to switch gears to dengue. I know I only have a couple of minutes left, so I won't go through why dengue is a huge, continues to be a huge burden right now, mostly in the developing world. But I would argue that with climate change, you can start to see areas of Florida, areas of Texas, and even where we are, areas of Southern California, kind of starting to get encroached with dengue virus. And so I was telling you about RDRP. So we took the same strategy we did for the nukes, screening the nuke library at Caliber. It's run by Sarah Cherry in collaboration with the folks at Caliber. And our current lead here is active in nanomolar concentrations against all 4 strains, Zika and yellow fever. So it looks like a broad spectrum flavy. It looked very well tolerated. The PK isn't great, but we're gonna try some in vivo proof of concept, and then also work on optimizing the PK. Hopefully, we can get an improved PK so that we can really establish a nice broad spectrum flavivirus inhibitor. Right now, there are no RDRP inhibitors for dengue. Janssen has a really good NS4B inhibitor, but we think that, like with all viruses, cocktails are going to be the way to go. So we're looking to combine that with the Janssen or other drugs. So speaking of NS4B, we are working in my lab on this compound C29, which was identified through a high throughput screen as a Zika inhibitor. Zika is probably not the most highest of unmet needs right now, but we through resistance studies, we found that it targets the NS4A protein. So what we tried to do was to look at some derivatives and see if we can get activity on dengue. And so what we found is that one analog inhibits Zika and dengue, and the other analog inhibits dengue. And when we did the resistance study on dengue, what we found was it hits NS4B and not NS4A. Of course, those two interact. So maybe, we're looking at, and it's not the same places that the resistance mutants that the J&J compound hit. So we may be disrupting kind of a protein-protein interaction surface here, but we're working on this and working to optimize it. But we have relatively good potency for this molecule on dengue, so we're going to try to optimize that further and move it into in vivo proof of concept hopefully by the end of the funding period. And the last program I'm going to tell you about is something that we found based on the fact that baloxavir works on other cap snatching viruses, including Lassa and SFTSV. Obviously, not at levels that can drive a repurposing play. And so the idea was to make baloxavir analogs that were bespoke for Lassa. What we used was LCMV here because it's not a BSL-4. And so what we found was that we found a number of analogs that are actually almost fortunately happen to be outside of the Roche Shionogi patent space, which, for Lassa doesn't really matter, but, still good to have freedom to operate. So we don't have a pharma company breathing down our necks, asking us why we're doing this. And these also have impact on Lassa. They're a little less potent on Lassa than they are on the LCMV. And so we're going to do a little bit more SAR to improve the potency on Lassa and then move in vivo for in vivo proof of concept. But with these numbers and with the PK we have, I think that we're pretty optimistic that we may have a nice baloxavir capsnatch inhibitor that will work for Lassa. The nice thing about Lassa is while oral is great, this is something that we believe we can also give IV, and so maybe the bar is a little bit lower for this than some of the other inhibitors that we're working on. So with that, I want to thank the team of folks that I've been working with this. Just, I think, a dream team of folks. Arnab and Adolfo are my co-leads, and I won't go through this list here. And also thank you for your attention, and happy to take any questions.